Good evening, ladies and gentlemen. Good, thank you. So I'm mic'd up and I'm just impressed by how loud my voice just got. Welcome to the University of Bristol this afternoon on what feels like the first day of summer even, and thank you for being inside out of the balmy spring summer weather to join with us in what is a special day in any professor's life. It's our chance this evening to hear what Jack has to say about the subject on the screen behind me. Let me start by introducing myself. My name's Guy Orpen. I'm the Deputy Vice-Chancellor and Provost at the University of Bristol. This is old stamping ground for me. I was explaining to Jack that many moons ago I gave my inaugural lecture as a Professor of Chemistry in this lecture theatre, and I'm empathising with Jack's challenge this evening. It's an interesting challenge to welcome members of your family, who I know they're clustered in the back row. Welcome, and thank you for being here. You're speaking to an audience of different levels of expertise in your field. I was speaking to members of my family who knew absolutely nothing of the wonders of crystallography, as well as the dean of my faculty, the pro-vice-chancellor for such and such, and lots of sceptical colleagues who wondered how this had ever come about. But that was just me. It's perfectly obvious how this has come about. Don't laugh at my jokes like that. It's inappropriate. So my job is to introduce the star of the show. It's conventional to say a few things about the story that got Jack here, or the professor here. I'm going to do that very briefly because I think Jack will spend some time reflecting on his own career trajectory. So I have a skeletal amount of information which I've just augmented through a cross-examination of Professor Mellor. It turns out that, after being born and raised in London to mathematician parents who are here present, Jack had, I'm sure, an excellent school career, but he doesn't mention here. And of course, he applied to the University of Bristol as an undergraduate to study psychology. Unfortunately, as was often the case back in the day when other people were running the university, we turned him down, and without interview even. So there you go. Things are much better now, as I say. But happily, Jack recovered from that and went to the University of Cambridge, which is an excellent university, so I'm told. I'm a Cambridge graduate myself. It is very flat and cold over there, but the university is very strong. And he successfully navigated the wonders of natural sciences at Cambridge, and then a PhD in neurophysiology, which has taken him down the path on which he still travels today. He spent 2.5 years in San Francisco as a postdoc thereafter, and then came to Bristol as a Wellcome Trust Fellow in 2002, and has been at Bristol ever since, rising through the ranks of various sorts of fellowships, then lecturer and reader. We didn't touch on that, but I'm assuming that was something in the middle there, or senior lecturer even. All those things. Risen through the ranks, up the greasy pole to arrive at the rank of professor, having been promoted about 18 months ago to this rank. So a stellar career that we're going to hear about in a moment. One anecdote that I promised Jack I would talk about was a dinner, I'm going to say 4 or 5 years ago, something of that sort, at which he and a number of the other audience members here present told me about what they thought neuroscience in the University of Bristol could be about, from a relatively junior position, senior lecturers, readers, that sort of thing. And as I say, a number of those people are still in the room. I have to say that was one of the most enjoyable dinners I've had with academic colleagues in this university. I got a very powerful sense of future leadership and ambition, and not in a selfish, individualistic way, but in a collective and ambitious way. And that made me very proud to occupy a senior position. I was then Pro-Vice-Chancellor for Research in the university. And I thought this is what makes this university great, that kind of ambition, that collegiality, that willingness to reach out to partners in the academic endeavour. So I would say that not just Jack, but others in the room and those not in the room today, thank you for that little experience, which meant a lot to me. I don't know whether it meant much to you, but it was certainly a very important and interesting experience for me, and it's echoed on, and I'm delighted to see the progress made by those same individuals and the strength that they are now bringing to their own careers, but also to the wider university endeavour. So without further ado, it is my very great pleasure to introduce Professor Jack Mellor to give us his inaugural lecture. Jack.
Thank you very much, Guy, for the introduction. It's very encouraging to hear you say those words about the meeting, because I think it was really my experience as well that was a really interesting and thought-provoking meeting, which really showed how collectively, as a neuroscience community here, we can achieve so much more than the individual parts often. And I think we're still moving towards that, but we still really are trying to achieve those sort of ambitions. So it's really great to hear that. I'd like to thank all of you for coming as well today, because obviously we're sat inside here and it's a rather nice day outside, so I'm sure some of you might rather be outside, but thank you all for coming as well. So I actually thought I'd split up the talk today into a couple of parts. The first was just to give an introduction to my background and how I arrived in Bristol. And Guy has given a short introduction. I wanted to embellish that a little as we go along. And then also to describe some of the scientific questions that we've really been interested in since I've been in Bristol, and to give you some sort of flavour of what that's been about. So Guy mentioned that I was originally very keen to join Bristol, but was unfortunately rejected. And I think actually that shows quite how insightful the people at Bristol were, perhaps. That actually perhaps they knew things that other institutions didn't know. And I guess when you see the person I was back then, the schoolboy I was, you can perhaps see a little bit more why Bristol thought that maybe I wasn't going to be the right person to be here. Is there anyone who can't identify which of those I am? Has everyone got me? Not quite sure? Well, to give you a clue, this is my sixth form class at school. I'm the one without the jacket and tie. I have to say, I did make quite an effort to spruce up for this photo. We knew it was coming in advance. And actually, normally my hair at that stage was down to my shoulders or below, and I'd had a rather nice bob cut to get me in the mood for this photo. But anyway, my idea of sprucing up, I guess, was to put on a roll neck jumper or something like that. But actually, the main reason for showing this photo is to highlight the role of the man in the middle at the front, who was my maths teacher for 7 years at school, actually. And he was a really inspirational teacher. He was almost completely unfettered by the necessities of the curriculum. He actually just wanted to teach maths and to teach maths to people who enjoyed it. And I think that was a wonderful experience for me. We spent hours in his classes just free-thinking, abstract problem-solving, which was absolutely brilliant for me. I think he couldn't care less about classroom management, so he therefore only really ever taught the top sets. And it was great fun, and I had a fantastic time with him. And I think part of that sort of academic freedom that I experienced there led me to think a little bit beyond where I was immediately at school. And so I spent quite a lot of time thinking about what I wanted to do after school. And I thought one of the most important sort of questions that faced me as an individual at that point was what actually determines human behavior, human motivation. And that actually made me think that what I wanted to study at university was psychology. So hence my application to Bristol to study psychology. And thankfully, they turned me down. I ended up at Cambridge, where you can't do psychology. You have to do a broad natural sciences degree. And so I only actually got a chance to do psychology. I hadn't done any at school at all in my second year. At which point, it dawned on me very rapidly that actually me and psychology really didn't work at all. I was hopeless at it. I think I got a low 2.2 in my unit in my second year. It was a disaster. And I think it was well, I'd like to think it's because my my sort of physical sciences, mathematical background really just didn't quite tie in with the kind of thinking that psychologists went through. So anyway, I ended up doing a neuroscience specialty degree, and loved it so much that I stayed on in Cambridge to do a PhD. So I ended up at the Laboratory of Molecular Biology in Cambridge, the LMB. This is the building. I was somewhere down in the basement, so you can't actually see the lab that I was in. Yeah, it was a wonderful place to be. It's a place made famous by Watson, Crick, and Franklin for the discovery of the structure of DNA. It's stuffed full of Nobel Prize winners. You can't move in there for bumping into them. Great place to do your PhD. It turns out that 5-10 years ago, Cambridge and the LMB decided that this building was not fit for purpose anymore. It was too old. You can see by the cars out the front that it was built in the 50s, 60s. And so what they did was they built a fantastic new building on the Addenbrooke site in Cambridge. Anyone who's been there will know that it is an absolutely amazing facility. You'll also notice from the architecture that the vintage of the building, the original building, is very similar to the building that we currently occupy here in Bristol, just up the hill. And I do actually remember Guy a few years ago saying that the building was no longer fit for purpose, and that we would have to rehouse everyone fairly soon. So anyway, we're anticipating the ambition that you expressed initially, that we are going to have a new building before too long. So yes, I turned up in the lab in the LMB with this guy who I see is here today. Where are you, Andy? There he is at the back. So Andy Randall was my PhD supervisor. He was a very junior group lead at the time, just starting his own lab. I think he was still in his 20s at the time. And Andy was a fantastic supervisor. He spent a lot of time with me, teaching me the dark arts of electrophysiology. And it's notable that everyone who's been through his lab, and there have been many of those people, I was, I think, his first PhD student, but there are many since, have all come out with a really excellent training in electrophysiology. I think it's also fair to say that I didn't spend all of my time in Cambridge at my PhD just working on science. I confess that I spent a good 40 hours of every week training out on the river rowing. I started off loving rowing as an undergraduate, and actually as a PhD student, I decided that what I really wanted to do was try out for the university team. So this is one of the crews I rowed in. It was a fantastic experience rowing. I spent a lot of time doing it. And I was reminded the other day that, watching the boat race, you always hear the commentators talk about how after the finish line, either they're slumped over their oars in despair, or it looks like they could row another 1,000 miles. And actually in this race, you can't necessarily tell whether we've won or lost there. Half of us seem to be completely dead, and the other half are still kind of pumped up. I think actually this reflects the fact of who was actually doing the work in the boat, but there you go. So, and actually I was reminded the other day of a conversation I had with Andy, and I can still recall it quite vividly. It was a few weeks before I started my PhD. I thought I'd better make him aware that I was thinking of spending quite a lot of time on the river, just in case he had a problem with that. And so I called him up and I said, Andy, I'm thinking of trialing out for the university rowing team, and it's going to mean I'm going to have significant time commitments. And he said, well, that sounds all right, but what exactly are these time commitments going to be? And I said, well, you train every morning before work. It's about sort of 2 hours, 6 to 8 in the morning. And then every weekend, we'll spend a whole time down the river. And there might be 1 or 2 afternoons during the week where we have to, go down the river. And he said, well, that sounds okay. We'll have to manage the time commitments and see how it all goes. It worked out. But I have to say, I put down the phone at the end of that, and I thought, I haven't been entirely truthful with you on that one. Because I think I did spend around about 3 afternoons a week down on the river. Anyway, I had a very successful PhD. I had a very enjoyable PhD, and it left me in a position at the end of it where I thought I really did want to stay in science. And I had an opportunity to go and do things, pretty much anywhere in the world. And again, I didn't necessarily make these decisions entirely based on scientific rationale. I kind of had enough of rowing by that point, but what was kind of starting to be one of my joys was actually surfing. So I sort of thought, well, where in the world could I go that would be really good to go and learn how to surf properly? And so, of course, the obvious answer was California. So as Guy said, I went to San Francisco to do my postdoc, to spend my time surfing at Ocean Beach. And again, I used to get up in the mornings, dawn patrol, down at Ocean Beach, surfing for an hour or two every morning before going into the lab, which was great fun. And the lab in San Francisco, I worked in the lab of Roger Nicole. And so Roger was and is a fantastic scientist. He was a hyper-competitive, very, very driven person who really was passionate about trying to understand how it is that the molecular machinery that enables synapses to change their strength, which is what we think underpins memory processes. And it was there really that I guess I've gained my core sort of ideas of what I wanted to study for the rest of my career so far, thinking about these mechanisms that occur at synapses. And so once I was finishing in San Francisco after 2.5 years, I was kind of thinking, well, I had such a great time in San Francisco. What I really want to do is recreate that experience back in this country, in the UK, because I knew I always wanted to return here. So I was trying to think, well, where's going to be the best place to do that? And the obvious answer was here in Bristol. And a number of reasons for that. I mean, obviously, you've got a West Coast town with a very iconic suspension bridge. You know, tick that box for Bristol. You've also got famously steep streets, which people love to race around. This is a picture from the film Bullet. It hasn't come out terribly well. But anyway, that's Steve McQueen racing around in his car in San Francisco. And for those of you who've seen it, in Bristol, they run a cycling race every year where they speed up Park Street as fast as they can. So the winning time is around about 20 seconds. So if anyone can think about that. But this is just a picture of the race up Park Street. It's called the Red Bull Hill Chasers or something, I think. Anyway, it's great fun. The other thing about this common between the two cities is a really great and vibrant social scene, a culture in the city which is really quite amazing. This is actually San Francisco. It could be any weekend in San Francisco, actually. But anyway, so I arrived in Bristol, and in all seriousness, there was very good reasons for me to come to Bristol, scientifically as well. And that was mainly about that Bristol is a world-leading centre in the study of synapses. And that was largely due to this guy and many others, but Graham was the guy who led up the centre here in Bristol. But for reasons I'll explain in a moment, I didn't come back to work directly for Graham. I actually came to work for this guy, John Isaac, as a postdoc briefly in his lab. And so John actually was a fantastic guy. Part of the reason I went to work for him is because he loves surfing as well. But he also had been a postdoc in Graham's lab and was recently a new group leader himself. And actually, interestingly, John had also done a postdoc in San Francisco with Roger. So you can see there's a link coming on there. And of course, Graham's first PhD student was Andy. And I was Andy's first PhD student, which in some ways, if you like these kind of things, makes me Graham's eldest grandchild, scientifically, which is a slightly odd concept to think about. But anyway, so what I'm painting here is a rather sort of cozy, incestuous, white male family environment, which is slightly uncomfortable, and I think in many ways actually, unfortunately, rather reflects academic science in many ways. Anyway, I'll leave that thought there, I think. So I should say that, so when I was coming to Bristol, in some ways that looks like a very cozy picture, but actually, as with many families, it's not all as happy and happy families as you like to think. And in fact, that was somewhat due to the fact that Graham and Roger were both very competitive and in the same field, and therefore they had disagreements. And often those disagreements might have spilled over into personal sort of animosity, shall we say. And perhaps this wasn't helped by, to some extent, my involvement in this. So I'm going to tell you a little story about what happened just before I arrived in Bristol to illustrate this. So Graham and actually Zunna's in the audience here. Hi, Zunna. Graham and Zunna published this paper in 1999 arguing that kainate receptors, which are a type of glutamate receptors, are involved in synaptic plasticity. And in San Francisco, in Roger's lab, we saw this paper and we thought, no way, this is not true. This cannot be right. We've got loads of data that argues that this cannot be true. And so we wrote to Nature, this was published in Nature, we wrote a brief communication to our Nature, which argued various scientific points. I've highlighted the important bit in yellow here. It says that here we argue that the available evidence does not support a major role for kainate receptors in the induction of multifiber LTP. Essentially saying, Graham and Zunna, you're wrong, and we're right. And of course, Graham and Zunna wrote back, that's the article down the right here, saying, no, we're right and you're wrong. And of course, I mean, as with so many scientific arguments, actually both sides were wrong, I think. Zunna, you may disagree with me, but and I think indeed, actually, we being Roger, myself, and Dietmar Schmitz, our co-author, published a paper a couple of years later, actually showing that indeed kainate receptors were involved in synaptic plasticity, but just not quite in the same way that Zunna had proposed. So, you know, there were some arguments to be had there. And I think I'm pointing this out in that in some ways, when I arrived in Bristol, I wasn't quite sure of the reception I was going to get, because actually I'd been part of a very public sort of argument about people's data from people who publish stuff in Bristol here. But I'm pleased to say that we're friends, and I'm still here. I'm not dead. So things turned out pretty good in the end. Okay, so I wanted to move on and just talk a little bit about the kind of scientific questions that we've been interested in since I've been in Bristol. And that's really in two parts I'm going to talk about. The first bit, I'm going to talk about how we think memories are formed and consolidated, and then also what determines, in the second part, which events are actually transferred into memory. So the idea that of how memories are formed really goes back to ideas from Donald Hebb and others many, many years ago, where they proposed that memories are formed by the association of different elements of an event, and that the brain might be able to actually do that if individual nerve cells are able to connect up in a stronger manner together and therefore associate different elements of that event. And so to illustrate this, I thought I'd just use the example of us all sitting here today. So if you imagine that inside the brain there's a dense network of nerve cells which are all interconnected, and each of those cells might encode or be active in response to a different feature of the situation that you find yourself in. So, for example, the red cell there in that network might be me, standing at the front here, talking to you all. Whereas the yellow one might be your neighbour who's sat next to you. You're experiencing that person. And perhaps your neighbour smells a bit. I don't know. Probably not, but maybe. And so you're experiencing that sensory input as well. And also, of course, you're also in this lecture theatre in the chemistry department, and maybe the ones of you who are really aware of what goes on in the chemistry department might know that this is a fairly high-risk environment for fires. And so you might be feeling a little bit apprehensive about the fact that there's a significant risk of fires in this building. I would say, and I had a chat with Guy about this before, that it also means that this building is one of the safest for fires, because everyone is hyper-aware of the risks involved. Anyway, so you might be aware of all these different elements of your situation at the moment. And the fact that that is the case means that all these cells will be active at the same time in your brain. And what Donald Hebb proposed is that because those are all active at the same time, what happens is that they all link up together in a much stronger way. So they form, if you like to call it, a memory ensemble or assembly of these neurons. And so that means that if, say, tomorrow you're out and about on the street and you happen to smell again the odour of the person who sat next to you, just say, you might actually then have that set of nerve cells reactivated, because the individual one here that is active by the smell will activate all those other ones around it. And so therefore your memory will be triggered of the experience that you had here today. So that's the theory that they proposed. But in order for that to be correct, there are a number of things that need to be in place. One is that you've got this dense interconnected set of nerve cells, and we know from the anatomy of the brain that that is broadly true. The other is that each one of these individual nerve cells needs to be selectively activated by specific features of your environment. And the final one is that you can actually increase the strength of those connections between your nerve cells by those cells being coactive at the same time. And that final point about the coactivity inducing a synaptic strengthening is something that was discovered partly in Bristol and partly elsewhere for a number of years ago, and it's something that's been studied very prominently here in Bristol by a number of the people in this room, in fact. And so I'm not going to go into this in detail, but broadly the mechanism that we study is the way in which individual molecules, if you like, are activated in order to cause a strengthening of the synaptic connection, the junction between nerve cells. So what happens at the junction is that electrical signals are found up here, and they then cause the release of neurotransmitter, and that then is detected by receptors on the second cell in the chain. And those receptors will then transmit an electrical signal on into that nerve cell. And there's two ways in which you can increase the strength of this synapse. One is to increase the amount of transmitter being released, and the other is by increasing the number of receptors here. There are other mechanisms as well, but those are the principal ones. And so people have studied these processes for a long time and broadly looked at how you can increase the number of receptors here. And we and others here have studied this for many years. So part of the reason for showing you this is that, therefore, one of the key requisites for that model I showed you originally is that you can get a strengthening of synapses, and this is indeed true. The other requisite I talked about is that each of these individual nerve cells actually responds to a specific element in the environment. And a number of people have actually shown that this is also true. So one of the famous examples of this is a study done by some researchers in the States where they recorded from individual nerve cells in a human brain. And so these participants were then shown a series of images with various famous people or famous places or animals or anything really. And so what the data I'm showing you here illustrates is the activity of a single nerve cell that they made a recording from in response to each of these different images. And you can see that these blue dots are the spiking activity of those individual cells. And what you can see here is that the spiking activity is very specifically in response to images of the actress Jennifer Aniston. And it seems to be that this was very specific. It didn't respond to other famous actresses. Indeed, it didn't even respond when you showed an image of Jennifer Aniston along with her, at the time, husband, Brad Pitt. That's shown up here. So it's just Jennifer Aniston herself. And I should also say that various other cells in the brain were responding to other different famous people or famous places. So what this really shows you is that there are specific images or elements within an environment that each brain cell is responding to. Now, this kind of study was only able to be done because these people were about to undergo surgery to treat their epilepsy. And the reason they had electrodes placed within their brain is to try and find out where that seizure activity was coming from so that you could take out the specific part of the brain that was required. And that means that this kind of experiment is actually incredibly difficult to do. It's very difficult to get the number of people you need to do a big study like this on. And that's why, for many people, what we do is to study these kind of processes in rodents. And actually, what we find is that in rodents as well, there are cells, particularly in the hippocampus, which respond specifically to different features of the environment. And somewhat famously, this has been shown that some of these cells can respond to specific locations when the animal is navigating within a circular environment like this one. So what I'm going to show you is just a little cartoon of a rat running around a circular maze. This is courtesy of Andy Doherty, actually. But on the bottom left there, you can see the activity of a single cell in the hippocampus as the animal's running around this environment. And you can see that that cell is really only spiking when that animal is in a particular location in the environment. So it's a very selective feature that the animal is responding to, or at least that cell is responding to. And I should say that there are, when you record from these animals, there are many other cells within the hippocampus that respond to different locations within that environment. And so the idea then is that by having multiple different place cells that respond to different locations, you will actually build up a picture as the animal runs through an environment. It will link up these place cells to each other by this process of synaptic plasticity to form a network of cells that encode your environment, encode the location within which this animal is exploring. But that's the idea. What people haven't shown or hadn't shown previously was that this actually occurs in reality, and that this kind of spiking activity could create these assemblies of place cells. And so this was a question that we set out to try and explore some years ago. And the way we do this is by recording from individual nerve cells within actually hippocampal slices. So we take slices of rodent brain to do this. And so we're recording from two different nerve cells in this slice, and you can see a fluorescent image of those cells below, showing the dendritic tree. And so if you actually record from both these cells and they are synaptically coupled, then if you stimulate the red cell on the right here with an action potential, it will fire its electrical signal, which you can record in the second cell here as the electrical response to that stimulus. And so you can measure the strength of your synaptic connection by the size of this electrical response here. And so we did this in an experiment where we, first of all, recorded the amplitude of this synaptic response. This is amplitude on the y-axis here. And then, after a little period, we replayed the activity of two place cells. That, as you can see, this is a representation of the place cell firing within a circular environment. They have very specific firing locations, but very much in the same location in the environment. And so that means that they'll be active at the same time, and you would expect them to induce some kind of potentiation of the synaptic strength there. But actually what we found is there's nothing at all. There's no change in the synaptic strength. And interestingly, what was required in order to get that change in synaptic strength was the presence of acetylcholine, which is a neurotransmitter released in the hippocampus in response to arousal or attention, or that's what it's believed to be responding to. And so what this suggests to us is that actually, in order to create memories or to form new memories, what you need to have is the release of acetylcholine. You need to be attending, you need to be aroused enough to generate the conditions required in order to form these new memories. And so we've spent quite a lot of time and effort finding out about the detailed mechanisms by which this happens. This is just to give you a flavor of some of the experiments we do. So the key trigger for initiating this process of synaptic strengthening is calcium influx into a synapse. And so in order to measure this, we fill a neuron. This is a nerve cell here, and here we're blowing up just a small part of the dendritic process. And you can see that up the top here. And hopefully what you can see there is that along this process, there are these little protrusions coming out, and they are the individual synapses that are made onto this nerve cell. And if we scan our microscope very fast across that individual synapse, we can then stimulate it and see, I hope you can see here, a flash, a spark of calcium influx coming into that synapse. So we can measure this in real time using this kind of technique. But actually, even with the best microscopes in the world, the level of resolution is at the level of this synapse. You can't go into the synapse and see in greater detail where that calcium is coming from. And in order to do that, what we did was to create a computer model of the synapse. and this is shown here. So this is the synapse sort of here. It's actually joined to a dendrite, which would be down here by this very thin neck, and that's quite common for synaptic processes. And so if I show you an illustration here, what we can do in this model is to stimulate the synapse as it would be in reality, and we can then predict what the calcium rises is within that synapse. So you can just see there that the calcium comes in at particular points in the synapse and then spreads across the synaptic process. So using these techniques, we've developed quite a complex picture of what goes on within a synapse in order to generate plasticity. And this plasticity is termed LTP, long-term potentiation. It's a term which means that the synapse will be strengthened. And the key trigger, as I've said, is calcium coming in, and it comes in through this protein here, an NMDA receptor, and also voltage-gated calcium channels. But the key molecule here is this one here, the SK channel, which is present in synapses and actually acts as a negative brake on these NMDA receptors. And so what you've then got within the synapse, I should also say these SK channels are activated by calcium. So you have this little regulatory loop going on within the synapse where the SK channels are critical for controlling the amount of calcium coming into that synapse and therefore controlling whether or not you generate any synaptic strengthening. So that's very interesting. The really critical part is that the role of acetylcholine, which is released, if you like, during arousal and during wakefulness. And it will act on these muscarinic M1 receptors we found to actually inhibit these SK channels. So, of course, if you knock out that regulatory SK channel function, then these things go crazy, and you get a whole lot of calcium coming in and the induction of synaptic plasticity. And so what this means is that, what it seems to mean is that you need the release of acetylcholine and that the arousal necessary to do that in order to generate plasticity and to form new memories. So the other thing that needs to happen in order to keep your memory is that it needs to be consolidated. And there are ideas now that the behavioral state in which that happens is during sleep. And so what is thought to happen is that you create this assembly of place cells during exploration when the animal's awake, and then you consolidate it by replaying that activity during sleep. It's almost as if you're replaying the activity that you've experienced during the day when you go to sleep. And so what people have found is that when the animal's sleeping is that this sequence of place cells that would have been activated whilst the animal is running through its environment is actually then replayed during sleep. And we had the idea that this replay was going to be important for further induction of synaptic plasticity, which would consolidate the memories. And so we tested this again by replaying the sleepy replay into our slices and found that indeed this replay event, which is shown by the arrow here, could induce a further strengthening of the synapses. So interestingly, we also found that if we blocked metabotropic glutamate receptors, that this synaptic strengthening doesn't occur. And so this then leads us to update our model where we have these metabotropic glutamate receptors, which are also present at the synapse, and can also, by a similar method to these, inhibit these SK channels, and so unlock the potential for plasticity. Okay, but the key part of all this is that, and I want to come back to it again, is that the release of acetylcholine is what's critical for initially forming those memories. And so what I want to come onto in the last part of the talk is the idea that when actually acetylcholine is being released will determine when we actually can form new memories. And so here we have the cholinergic system within the brain. This is the basal forebrain, which has most of the cholinergic cells, which project out to the whole of the brain. And surprisingly, when we started out on this project, there was really very little information about when acetylcholine was actually released. And this was doubly surprising in some ways because the major drugs which are given out to help people with the memory deficits found in dementia and Alzheimer's disease are these drugs here, which are anti-cholinesterase inhibitors. So they actually boost the level of acetylcholine in the brain. And so it's really surprising that we don't actually know what acetylcholine release they're supposedly boosting. So we set out to try and find this out by using some choline sensors which were designed and developed by John Lowry, who's a guy who works in Maynooth University in Ireland. So very briefly, they work by having an enzyme, choline oxidase, embedded in a polymer around it that binds to acetylcholine and releases electrons which can be detected on the electrode. So you can measure acetylcholine release by a current flow. And so we implanted these sensors into a mouse brain, and this is what you see. So you can record continuously across a whole day. This is a whole day's worth of recording here. And you can see that, first of all, when we put the mouse onto a maze, the level of acetylcholine goes up quite dramatically. And then when we put it back into its home cage afterwards, the levels go down gradually. And when it actually falls asleep, you can see that it's really quite low, but there are these sort of short, small little blips in the acetylcholine levels. And these seem to be associated specifically with going into REM sleep. So we have no idea what that's about. I don't think anyone really does understand why acetylcholine is higher during REM sleep. But that seems to be what the result we have. But broadly from this data, we can argue that acetylcholine seems to be involved in arousal processes. So when the animal is having to do something that's cognitively demanding. We also found that there are these very fast acetylcholine release events, which are on a timescale of seconds now rather than many minutes. And these seem to only occur when the animal is trying to perform this maze task. So we then looked at when exactly these acetylcholine release events are occurring. And so this is the maze task that the animal has to do. It starts here. It gets up to the choice point here, and it's forced to turn either right or left, at which point it gets a reward at these corners. It then returns back to this zone and is held there, and it has to remember which way it was told to turn initially. Then when it runs up the second time, it has to remember to turn the opposite way this time, and then it will get a reward again. So when we then analyzed our data to see when these acetylcholine release events were occurring, we found that they're almost predominantly in the reward areas. And this actually was very surprising to us when we found this. No one had ever shown this before. But in retrospect, maybe it's not quite so surprising, because if we think about our model whereby acetylcholine is allowing the encoding or the formation of new memories, what you really want to be able to do is to learn from the outcome of your experience to form the new memory, and to therefore make better predictions about your decisions the next time you come to them. And so actually having acetylcholine released at the reward zones makes some sense in some ways. And I think this has then led us into a sort of a new territory for us in many ways, that what we're now thinking about is why it is that you would want to learn something new to update your internal sort of representation. And so ideally what you're really trying to do all the time is to make better predictions, and therefore to enable you to make better decisions. And I guess a good example of this recently is the fact that the recent presidential election, everyone was predicting, up until just a few days before the election, that Hillary Clinton was going to be the president. And in fact, the reality on the day is that we had President Trump. And I mean, even to the extent where these magazines were making up headline pages on two different, immediately on their realization that they got it completely wrong. So the expectation, if you like, in this instance was completely at odds with the reality on the day. And at that point, you'd say you have to really learn something new. And in many ways, we're still learning to update our expectations based on this outcome. So and I'm nearly done, don't worry. But I wanted to sort of talk a little bit about where we're sort of going with all this, and to put together a sort of framework, a schema for how we think the memory process might actually be working in the brain. And this is really all to do with this idea of creating or resolving uncertainty within the brain. And so if we think about uncertainty as being the result of entering an environment which we don't know the outcome. So maybe that's a novel environment, a new environment, or maybe it's being surprised by receiving an outcome that you weren't expecting. And that will increase your level of uncertainty because you no longer can make good predictions about what's going to happen. And so what we propose is that will actually cause the release of acetylcholine and also increase your attention and arousal levels. And then, because of the work we've done, we predict that will allow synaptic plasticity to occur, and therefore we'll be able to update our expectations by learning about the new things that we've seen. And this ability to update will then reduce our levels of uncertainty, because now we know much more about the environment that we're about to go into. And so what we've started to think is, well, what happens if actually you have problems with this process here? If synaptic plasticity goes a bit wrong, what's that going to lead to? It means you can't update your expectations, and therefore perhaps your levels of uncertainty are going to keep rising. And that can lead to anxiety, certainly for one, but maybe other problems as well. An alternative way of thinking about this is that actually it doesn't really matter if you don't update your expectations, if you don't pay any attention to reality. And so famously, James Comey, I think, said last week that some people can be completely untethered to the truth, and that doesn't really matter too much. Well, that's a matter of opinion, but I think this illustrates how this is could be a really important process by which we're actually controlling our behaviors and thinking about how human behavior is motivated. And just to finish with that, and this is something that we've been thinking about, and partly as a result of my wife, who is a psychiatrist, and has got me starting to think a little bit more about how some of our work actually impacts on human behavior in mental health. And so there are many risk factors for mental illness. But one of the key ones is early life adversity. So traumatic or very stressful events early in life can lead, in many cases, to subsequent mental illness later on. The other major risk factor is your unique genetic makeup. And what we've been thinking is that both of these processes, there's very good evidence that they impact on the process of synaptic plasticity. And so that then this may then feed into this kind of schema, this framework that I've outlined, and lead to problems with dealing with uncertainty in what is often a very uncertain world in any case. So I'm going to stop there, but before I finish, I'd just like to acknowledge the many great people who I've worked with. And actually, when I was preparing this, I was reminded of a favorite phrase of my mentor, Roger Nickel from San Francisco, who often said that science is a human endeavor. And I think in the context he was talking about, what he really meant was that all our interpretations of our data and our scientific discussions are always based on often on our unconscious biases. And that's something that we always need to be aware of. But I think in this context, I often think of it as being that whenever we do science, it's always about the people we're interacting with. And what has been great in Bristol is that I've worked with a huge number of really fantastic people, both as scientists and as people. I hope I've listed as many of them there as possible. If I've missed you off, I really apologize. And very finally, I'd just like to acknowledge my parents who are here today, who really gave me an awful lot as a child and throughout my entire life. I think that support has been key in making me reasonably resilient, I think, which is a key aspect for an academic these days. And I think they've also given me a very healthy appetite for never accepting the received wisdom, which again is also something that's really key as a scientist. So I'd like to really thank them. And I will finish there, and if people have questions, I'm very happy to take them as well.
Normally we don't bother with questions.
That's okay.
Yeah, you're far away, and I'll finish up.
Thank you. Well, thank you very much.
Stand next to me, then you'll be recorded. Well,
I think I've got you.
Oh, you have it.
Thank you very much, Jack, really for an inspiring lecture. It's very difficult to, I think, sort of give a lecture that cuts across all sorts of expectations and areas of uncertainty that the audience has about understanding what you're going to talk about. We're a broad church of people here, so I think you did brilliantly in sort of making a complex subject easy to understand anyway. Now, I'm going to, I'm Christopher, I'm head of school here, where I have the honour to sort of work in a department where we have Jack, who is producing science such as this. Now, the reward for being here, in other words, an increase in your cholinergic mechanisms, will be some alcohol outside afterwards. But just to increase the uncertainty of when the reward process will take place, there are two aspects. Firstly, as Jack said, there's an opportunity for you to ask questions. So, does anybody want to quiz our star lecturer with a question that would, well, not exactly prove him wrong, I suppose, because Graham is not here to sort of do that. But does anyone have a question they would like to put forward? Yes, please.
How do you quantify uncertainty?
That's an excellent question. I think the short answer is we don't have a good mechanism of doing that at the moment. What we are proposing to do is to use what's termed a probabilistic rewards task. So essentially, if you have an animal in an environment where it receives a stimulus and it only gets a reward a certain percentage of the time, then that creates a degree of uncertainty for the animal, and we can regulate that degree of uncertainty by changing the probability of the reward that's given. So that's a short answer, and probably not a very good answer to your question, but that's the best I can do, I think.
Jeremy.
Sorry? Is smoking good for the brain?
No, certainly not.
Why do you ask, Jeremy? Yeah. So the mechanisms that we've talked about today are acting on muscarinic receptors, which are different to the nicotinic receptors. Interestingly, there's a model for cognitive deficit, which is in smokers which are taken off their drug, their cigarettes. So when you take people off their cigarettes, they become quite cognitively impaired, actually, for a while. So whether that doesn't really answer your question, but it does indicate that there are some cognitive benefits to smoking. Yeah. But the brain seems to adapt to that level of cognitive enhancement so that when you actually take them off it, they go below their normal performance. Yeah. Sorry? When they start their habit again? After a while, yes, they come back, yeah. Yeah.
Right. Saf, did you have a question?
Sleep is essential for consolidation of learning. Does it matter in terms of when you learn to take during the day? Is there time between learning and sleep?
I don't know the direct answer to that question. I'll tell you an anecdote instead. So I have this theory that, and as a parent of young children, that the chronic sleep deprivation that occurs when you have young children is absolutely crucial for the survival of the human race. Because if you could remember quite how painful that is in the early years, you would never have more than one child, which means that the human race would die out. That's not an answer to your question.
Yes, please.
A flippant answer would be maybe you need to get back on the cigarettes, but again, the answer, I don't really have a good answer for that. You've got a little block somewhere which isn't reactivating the right circuit exactly, and you need to unblock that somehow. Could be that the drugs will do it, but yeah.
A question, second row from the back, please. I'll come to you.
Does the size of the reward correlate with the intensity of the acetylcholine?
So we don't know the answer to that because we didn't give them different size rewards. That's something we're planning to do, and it'd be really interesting to find out whether or not that's true. We don't know at the moment. The thing we do know is that the level of uncertainty about whether or not that reward is given does seem to correlate with the amount of acetylcholine. So the more surprising the reward is, the bigger the level of acetylcholine, which fits quite neatly with this idea of uncertainty. Yeah.
Please.
Does that mean that once those cells, once I've allocated a genotransfer, these set cells and whatever, that's it, they're permanently that? So then what would be the implications for people perhaps who were born with perhaps lying or death, who later have those senses restored through advances in biomedicine? Those cells then be able to join in, or will this already be learned through childhood?
Yeah. A great question, which again I don't have a good answer to. I mean, one aspect of that is that once you're in love with Jenna Aniston, you're in love with her for life, right? But maybe not. But yeah, I don't know the answer to that. There is some evidence that you can change the responsiveness of certain cells to different stimuli. There's certainly a plasticity within the system which can change the actual, what each individual nerve cell encodes. But I don't know the details of that, and I certainly can't answer the wider aspects of your question.
Why the cell could be having its former association removed, something else occurring if needed?
Yeah, I mean, that does seem to be the case. Yeah, you can do that.
Zaf, you've had your hand up for a while. No, you haven't. It's not Zaf, it's Zuna who's going to ask the killer question now, aren't you?
Yeah, this is the one I've really been worried about. Yeah, having a nap during the day. Yeah, it seems like that's a great idea, doesn't it? I don't know, I didn't see too many people sleeping in that lecture, actually, but I think, so there have been nap studies, and I wish actually Liz Coulthard couldn't be here today, but she's done some work looking at the effects of napping on memory retrieval, memory consolidation. And actually she seems to show that, yes, napping is quite helpful. So, yeah, maybe we should all have a little siesta after lunch every day. Yeah.
Okay, there's a question. Time here.
We're deeply into psychological domain, which, as I've explained, I don't get on with terribly well. But, yeah, again, I don't know the answer to that question terribly well. But you can imagine that if you are thinking very broadly about different things and maybe increasing your levels of uncertainty and therefore potentially anxiety, that maybe that does happen. But I'm afraid I can't really comment too deeply on that.
Tony Pickering over there, top right.
Several thousand%, I would say, yeah.
Right. Right, we're going to stop questions there because Guy's getting a bit worried. But I'm just, as head of school, then, but I do want to not just praise Jack, but I want to praise the environment that we're in, and I just want to illustrate that to you, in as much as that early this year, or at the end of last year, we had the opportunity to appoint 2 new neuroscience lecturers in the school, which we did. We had a choice out of 89 applicants who originally applied. A rigid triage managed to reduce that to 86. And then the difficult choice of making shortlists and interview panels came up after that. It just tells you what reputation neuroscience has in the world by the applications that we got, because some of the people that we had were truly remarkable. We managed to appoint 2 people, and we're going to welcome those soon to the school. It will involve a little bit of expense in getting them into the building, but we have it from now the Deputy Vice-Chancellor that there will be no expense spared. And so we look forward to welcoming them to our new environment as well. So permit me one story about not recognising people. There's a story told by Lord Boothby. I don't know if you've heard of him, but he was a bit of a rake involved with the craze in London at some stage, but generally a fine fellow. He was wandering around Fortnum Masons before the war, and he came across this problem. You meet people, what on earth is their name? Can't remember. You have this facile conversation with them. He said to the lady, and your husband's still doing well? She said, yeah, still king. So clearly the cholinergic mechanism was lacking there. Ours will be rewarded now. There's refreshments outside. There's opportunity to speak with Jack afterwards again in a more sort of convivial environment. And I, on behalf of the school, on behalf of the university, welcome you all coming here as well. So thank you. Thank you.